So it's my pleasure to introduce the first session today. So it's appropriately entitled Transitioning from Academic Drug Discoveries to Biotech Clinical Trials. And as Stan was just alluding to, we really do have a great lineup of speakers here. And something that you'll see in common with several of the speakers, especially in this first session, is how they've lived both the academic and the industrial drug discovery and development process. And they've really come from different paths, and therefore they really bring a wealth of experience and expertise that they've kindly offered to share with us today. And we are not only grateful for having local speakers here, but people have traveled from across the country at the last minute, as Stan mentioned as well. And so with that, it's my great pleasure to introduce our first speaker, Michelle Arkin. Michelle is a professor of pharmaceutical chemistry at UCSF. She's also co-director of the UCSF Small Molecule Discovery Center. She was a founding scientist at Sunesis Pharmaceuticals before venturing off to UCSF and launched a very successful career there. She's highly experienced in the areas of chemical biology and drug discovery. She's a very busy person, to say the least. She's also president of the Academic Drug Discovery Consortium, represents UCSF in the National Cancer International Chemical Biology Consortium, and the ATOM Consortium. And ATOM stands for Accelerating Therapeutics for Opportunities in Medicine. So Michelle's topic today will be from basic science to drug discovery, pointers and prospects. And so if you're new to this space, be sure to have your pens and papers ready to go.
Thank you.
Thank you, Michelle.
So we're talking about the early stages of drug discovery, and I was having dinner with a colleague the other day, and he said, we don't call it basic discovery, we call it discovery science, because basic sounds like not as hard and challenging and exciting as basic science actually is, so I should change the title of my talk to discovery science, from discovery science to drug discovery, but it sounded funny. So you have some new biological discovery that you've made in your lab, and maybe it's understanding how a protein works, maybe it's understanding a new phenotype responsible for a disease, and you want to say, can I turn that into a drug discovery? Can I turn that biological knowledge into something that I can make a small molecule drug for, that I can give to patients? And that process, you'll see it, I bet, in every talk in some format or another. There's kind of a chevron of from discovering a target rather, starting a drug discovery project with target identification, and then ending up with a new drug application before the FDA, and then a pill or something, or an injectable protein, for example. So for small molecule discovery, we go through these multiple stages, and it's not really as linear as this. It's really a highly iterative process. But we start with the target, and then we find some compounds that address that target. If it's a protein that binds to the target, inhibit its function, for example, or if it's a phenotype that modulates the phenotype, optimize those compounds, and so forth till we get to the clinic. So basically, when we talk about early stage drug discovery, we're talking about target selection, hit identification, so finding the first molecules, and doing some initial optimization for cell-based potency. Then as you move further down the drug discovery pipeline towards the clinic, you're raising ever higher and more complex hurdles that your molecule has to go through. So in later stage discovery, some people call it preclinical development, we're optimizing the molecule now for animal efficacy, for the pharmacokinetics and pharmacodynamics. So what the pharmacokinetics, what the body does to the drug, degrades the drug. You don't want your molecule to be seen as food and get digested, or as poison and get excreted. Right? You want it to survive in the body to get where it needs to go. And then toxicity and safety is usually done fairly late in the process in IND-enabling studies, so investigational new drug enabling studies. Then we go into clinical development, and the 3 phases of clinical development start with a measurement of human pharmacokinetics and safety. So these are things that should be pretty well modeled by your early preclinical development. So we're doing dose escalation to see where the compound is toxic. Phase 2 is like a pilot study for efficacy, and then phase 3 is a registrational trial for efficacy. So what could possibly go wrong? So the beginning part before we're in humans is called drug discovery, and the part once we're in humans is called drug development. But the thing with drug discovery and development, and we all hear about how expensive it is and what an enormous industry pharmaceutical development is, and yet we have some years only 20 new medicines come in. Some years, this year we had a banner crop of about 60. So the reason that we have that huge industry and it costs so much money to make a drug is because there's a lot of failure. So if we want to make drug discovery cheaper, we just reduce the amount of failure we have. And that's what a lot of us are trying to do, right? We're trying to select better targets, more insightful assays, more rapid and insightful ways of doing drug discovery to bring the cost down and bring the failure rate down. But currently, the failure rate for new medicines is more than 90%, so 10% success rate in the discovery phase in pharma, and then about 10% in the development phase also. So after spending those years and that money and all that insight that you're generating about your target, it still fails 90% of the time. And for some therapeutic areas, like Alzheimer's disease, it fails 100% of the time. Okay? So why is that? Where do things go wrong? So in preclinical, once we get pretty close to the clinic, and we're just doing those our last final touches before we go into the clinic, 60% of the time, those projects fail for safety reasons, which makes sense. That's what we're measuring in IND-enabling studies. So 60% toxicology, non-clinical toxicology. But then when we get into the clinic, in phase 1, 25% of the time, compounds fall out for safety reasons. Okay? They can't be advanced any further because we don't have safety at the dose where we have activity. 15% of the time it's for pharmacokinetics, so we can't measure enough of the molecule, not enough of the molecule gets to where we need it to go. This is down from much higher numbers before we started doing these PK/PD experiments that your new core or your newly refurbished core is working on. Now we do pharmacokinetic studies very early in the process, and for that reason we have much less failure late in the process because we bake in those parameters early into our drug discovery process. So if you think about what makes drug discovery so hard but so fascinating, everything that's going to happen to that small molecule in the body, is in the commercial space, in the world, is encoded by 30 atoms, 30 to 40 atoms. All the things it's going to bind to, how long it's going to survive in the body, what other things it's going to bind to, what's going to make it toxic, is really encoded in a very small amount of information. And therefore, every little bit of that information is very important. Okay? So we try to bake in pharmacokinetics early in the process so that we have good exposure at the end. But then now why do we think things fall out in phase 2? More than 50% of things fail in phase 2 clinical trials. Largely for efficacy reasons. This 30% number varies by therapeutic area quite a lot. In cancer, it's much higher than 30% of the time. 25% of the time, it's not safe. So more than 50% of the time, we don't have a safe dose that's also efficacious. Okay? So there are two basic reasons. Either it's a wrong target or it's the wrong molecule. Other than that, it's great. We're in great shape. Okay. So we have to think both about the right target and the right molecule. So what can we do in the early space? Most of us, I mean, probably some of us are clinicians and development people, but my group works in the early space in drug discovery. So what can we do in the early space to try to reduce the amount of failure we have in phase 2 clinical trials? So I think of the problem in 3 basic bins. One is selecting the wrong targets. Okay? And why do we select the wrong targets? Because we don't validate those targets completely. Okay? We don't fully understand the role that that target's going to play in the biology and in the disease state. And second is that sometimes we do really know what the right target is, but we don't know how to address it with a small molecule. And so we call this undruggable, and we either look for something we do understand, a GPCR or a kinase somewhere near that target we think we're interested in because that we know how to drug, or we put it in a bin. Or we can say, all right, it's called undruggable because it hasn't yet been drugged. So what technologies can we bring to bear? What new ways of thinking about the problem can we bring to maybe tackle that so-called undruggable target? Okay, so sometimes the target we don't understand well enough. Sometimes we do, but we don't know how to make a molecule for it. The other problem, and one of the problems with target validation, is that we select the wrong model systems. And this was especially true in the earlier days of high-throughput screening, where in order to do something hundreds of thousands of times in a way that won't completely break the bank, we need to simplify. We need to miniaturize. We need to simplify. It needs to be robust enough to miniaturize that experiment and test it hundreds of thousands of times and believe the results. And there is maybe too much simplification. Okay? We oversimplified the system for the sake of efficiency and engineering, and then what we get at the end, because we get what we screen for, what we get at the end is not going to work. And you probably know also in your own therapeutic areas that animal models can also be a problem. If the animal model is not predictive and then we make decisions based on it, then we may pick the wrong molecules. Okay, so in high throughput screening or in the early stages of drug discovery, what can we do? How can we challenge ourselves to use state-of-the-art microscopy or state-of-the-art structure, these technologies that will allow us to really get highly insightful assays that are also affordable and quantitative. And then finally, there's what we mentioned, 30% fail for efficacy, 25% for safety. Let's think about that ratio very early in the process. Instead of focusing on affinity, the molecule's tight, tight, tight. Think about tight and safe, tight and safe. Always thinking about therapeutic index, so the concentration at which you have activity and the concentration at which you have a safety or you stop having safety. How big can that margin be? Okay? Because then patients are not as well defined as cells or as animals, and you have variability in there. So we want to have a wide safety margin. Okay? So to think about safety and pharmacokinetics and activity all at the same time, we need to really be working on parallel tracks mentally and experimentally, and that's challenging, and that's one area that artificial intelligence and machine learning can really grasp a lot of that, process a lot of that data and try to help us make better decisions about all three in parallel. Okay, why is target validation difficult? Why is target discovery difficult? Because it's very complicated. So this is a metabolic pathway. Maybe you guys have seen this. Maybe you have nightmares about it. So it's very hard to know, where do I want to be? What's upstream of what? What's downstream of what, right? Biology is very complicated, and it exists on several levels. And if you're looking at the wrong level of the system, then you might miss things that are really important. But it's really important to pick a good drug target, because it's a kind of a truism in the pharmaceutical industry that a bad target never leads to a good drug, but sometimes a bad drug can be really effective. Okay? So a bad drug meaning not perfectly clean, not exactly the pharmacokinetics you want, all these things. But if it's hitting the right target, then you can often turn that into a drug. Doctors are very good at figuring out how to handle certain kinds of toxicity, right? They're used to dealing with them. So a paper that I think is very nice, easy to read, easy to understand paper by Gashaw et al. From Drug Discovery Today in 2012 lay out what do we mean by target validation from a pharmaceutical company perspective. And so if you are thinking about doing drug discovery, it's useful to think about this, because these are your target audience, right? Venture capitalists and companies are going to think about this the same way. So basically, selecting a good target is an intersection of understanding how the disease works and how the target works in that disease. That's obvious, right? Understanding the disease, maybe understanding the mechanism, but also access to models and technology. So the ability to test things in a way that's really going to give you insight. So then breaking this down, start with an important problem, a disease with high unmet medical need. Identify a molecular target or a phenotype. So the history of drug discovery, modern pharmaceutical drug discovery, is very target-based, right? We purify a protein and we understand how the molecule binds to the protein. But I want to include phenotype, because a lot of academics and more in pharmaceutical companies as well, we're looking at complex phenotypes and trying to find molecules that modulate phenotypes rather than targets. But say we start with a target or a phenotype. What's known about the target? How likely is it to be druggable? There's that word again. How do we select patients and monitor the activity of the drug in those patients? Very early on. Thinking about that. Who am I going to treat, and how am I going to know that the drug is working in those patients before I just do it empirically and find out it doesn't work? What safety issues are likely based on the drug or based on the target, drug class or target? And then consider developability, and we'll hear much more about this from people who are more expert than I. But developability includes the freedom to operate, so the patent space, competition, and also something that crops up a lot, the common mechanism potential of the target. So what this means is that there are advantages in drug development in working with the FDA and in selecting patients to select a narrow patient population that has a very well-defined disease. But then that narrow patient population, you may not be able to develop a drug for them because it may not make enough money. It may not be worth the manufacturing. There may be too few patients. But if you can take advantage of the ability to get a drug developed with those well-defined patient populations and then transfer that mechanism to a larger patient population, then that's a way that a lot of pharmaceutical companies will say start with a rare tauopathy with the idea that they're eventually going to test all sporadic Alzheimer's disease, or a rare cancer with the idea that other patients and other indications are also going to benefit from that mechanism. So, I mean, I'm a chemical biologist, so I think you can solve all your life's problems with chemical biology. And so these are our 4 rules of chemical biology. So when I'm talking about chemical biology, I'm really meaning using molecules to do all that stuff that I was just talking about. So can you use a molecule to help validate your target, a pharmacological type molecule, a prototype drug, to tell you hitting this target is important, this animal model is predictive, these are the best cells to use. So to get insight into how that target works in biology, using a molecule that looks kind of similar to what the drug is going to look like. One thing to caution you, chemical tools, a chemical biology probe, to really give you insight into the biology needs to be much more selective than a drug may need to be. A drug needs to be not toxic at the dose that it's effective. Okay? A chemical tool needs to be specific enough that if I say I'm inhibiting MYC and I'm going to use that molecule to tell me all these things about MYC, that it is really MYC that I'm reporting on, okay? MYC inhibition that I'm reporting on. So the 4 rules of chemical biology start with an interesting and important problem. Make molecules that could address that problem. Note, you will do chemistry. You will do chemistry. You will not screen for your drug. You will screen for a starting material that you will do chemistry to become your drug almost always. I guess not always, but almost always. Prove the molecules do what they're supposed to do. Don't skip this step. Okay. Use the molecules to answer the question. So when we, especially in academics, when we do drug discovery, very often, and this is true in biotech and in pharma also, much of the time what we call drug discovery is really chemical biology. And it's okay. It's okay. It counts, because chemical biology is a really insightful way of learning about biology and getting you tools that are likely to turn into something. And the other point that's really important to make is that it's a team sport. Okay? Drug discovery is a team sport. So be nice. Wear a helmet. So let's talk just a little bit. Okay, now we've selected a target. We have an idea about what we want to modulate and how we're going to go about doing that. The next step is finding a hit, so finding that first molecule. And it really is important to start with the right assays. And assays can be widely variable, widely diverse. So high content imaging, for example, is really big on the scene now. Almost all the screens that we do in the high throughput screening lab I direct use high content imaging. So this is an example where high content imaging is just automated microscopy, sample collection and analysis being automated. Here we're looking at cilia, these little non-modal antennae on the surface of mammalian cells. You can't really see that any other way but by microscopy. We also can look at very diverse things. These are whole parasitic organisms that we can get into a dish. These are parasitic worms, and you can see they move. We can quantify how they move in response to perturbations like drugs or genetic manipulation. So we can get a lot of insight, a lot of diverse insight from high content imaging. But also just good old target-based discovery can also provide a lot of insight. Here we can think of it as a forward and a reverse chemical genetics. Okay? Forward chemical genetics, I'm going to get it backwards, is do the phenotype, find out what the target is, figure out how the biology works. That's a phenotypic assay. Reverse chemical genetics is find a molecule that binds to my target, see what that target does in biology. Okay? Both work. Both are important. So in your assay, you want to balance complexity to really capture the most important aspects of the biology, and also simplicity. You don't want to be more complex than necessary. You don't get extra credit for doing something more complex than necessary. Then you have to test the right compounds, and we'll talk a little bit more about that, but library design and different types of libraries is really important, and it's an art form. In fact, tomorrow, we're having an academic drug discovery consortium conference in Boston on new technologies, library technologies, trends in library design. And then finally, extract the best hits. So when you look for a molecule that's active in your assay of choice, not all compounds are active for the right reasons. Okay? So the most important thing is mechanistic validity, not potency, but mechanistic validity. Is it doing what I think it's doing in a way that's then optimizable? So we'll talk about that a little bit more. And then is it chemically tractable? Is it something that we can look at and say, oh yeah, I know how to make that. I can make analogs of that. I can encode all those different things I need to encode in my molecule by altering it through chemistry. This is what a high throughput screen looks like. This is just pulled a screenshot from our web system that we use at UCSF. And what you see is that most compounds don't do anything. So in green, this is actually a cytochrome, which is why for people who do high throughput screening, it's such a high hit rate. It's a metabolic cytochrome. But in green, we have 0% inhibition. Okay, so maximally active enzyme. In blue, we have minimally active enzyme. And then in red are all the test compounds. So what you want to see is that most of the test compounds don't do anything. They sit around the green line. And then some compounds do something. Here, if you click on one of these compounds, we can get the structure and a bunch of information about it. So one of the pieces of information I want to point out is if you look at, this is now the plate from whence this molecule came. Most of the plate doesn't do anything. Right? Only a few things on the plate are active. They're randomly scattered on that plate. This is a 384-well plate. And the Z-prime score, Z-score and Z-prime score are measures of how precise the assay is. Okay? They're not really measures of how good your compound is, but they're measures of how good your assay is to identify compounds. And a Z-prime score is the mean and 3 standard deviations in the negative controls, and the mean and the 3 standard deviations in the positive controls, and the distance between them. And then 1 is perfect, meaning infinitely small error, infinitely large range. Greater than 0.5 is considered excellent. When you're reviewing grants, it's not considered it's considered excellent is 0.5, and often you don't get that in high content imaging because the changes are small and the errors are big, right, because it's more complicated. But in a biochemical assay, you usually can get well over 0.5, and this assay is 0.9, so highly precise large window assay. So what's the goal of high throughput screening? It says at the top of the slide, it's to weed out the stuff you're not interested in. Okay? So I'm not interested in any of these. Some of these I will be interested in, and some I won't. And when I say that, I mean some of these are going to be highly reproducible, but not active for the reasons that I want the drug to be active. They're not active for reasons that are optimizable. They're going to lead to a drug. And this is, for those of you who know Roz Chast from The New Yorker, this is Roz Chast did this cartoon for Nature, for this Nature article in 2014. Where we're looking at different structures that show up a lot in high throughput screening libraries because they're easy to make, but they're highly reactive or have other things that make them what we call frequent hitters. Frequent hitters, bad apples, aggregators, PAINs, pan-assay interference compounds. There are a lot of different names for them. But they're molecules that show up a lot. And we really, when we sit on study section reviewing these kinds of grants, we really look at the structures of those hits and see, okay, are those drug-like structures? If they're not, has the lab proven that they're actually binding to the target of interest, that they're not just acting in this nonspecific way? For example, membrane disruptors, metal complexers, generate reactive oxygen species. Some of these are really challenging, and some are active in cells. Some are only active in biochemical assays. Some are only artifacts in particular types of biochemical assays. So luciferase inhibitors, for example, are very common in high-throughput screening libraries. It's a very druggable enzyme. So if you're using firefly luciferase, you have to be aware of whether those compounds are just hitting the luciferase. So the key thing, and this is one of the slides that is more helpful read than visualized, there are many things that you want to know about your molecule coming out of a high-throughput screen. Not just is it active, but is it active broadly? So it can be correct, and if it only works in one cell line under one condition at one time of day, it's not going to be a drug. So it has to be robust and broadly active in a range of different activities. It should be active in orthogonal assays, as orthogonal as possible. You want to make sure the compound doesn't aggregate. and pull the protein out of solution with it. Most importantly, I think what gets rid of a lot of these different problems is if you measure that it's binding to the target. Well, of course it's binding, not necessarily binding to the target in a drug-like way. So a drug-like way, in my world, is one molecule binding to one protein at one site. Okay, so a stoichiometry of 1-to-1, or one binding site per molecule. And there are many different ways that you can measure this. And some of these measurements are actually quite high throughput. You can even do a primary screen that way. Okay, then more details. You want to understand the mechanism of inhibition if you're inhibiting an enzyme or a G protein coupled receptor, binding stoichiometry, binding affinity, and then, of course, the binding site, high resolution structural biology. Okay, so many problem artifacts, many things you want to measure, and they all fit together into a workflow. Okay? So this is really the main point, is that you have some kind of primary assay. Every assay has some problem, something it teaches you, something it doesn't. And then when it's active in that assay, you go into a set of workflows, so a set of assays that you can do that ameliorate the problems that the other assay might have given you. So this is a biochemical assay. Now we look at the kinetics. How is it inhibiting the enzyme? We look at a direct binding method by NMR or surface plasmon resonance. We make sure that the compound is not aggregating by looking at dynamic light scattering. So these compounds will form aggregates, and you can see them scatter light. And then if you get through these filters, so it binds to the target, it has an inhibition mechanism that we understand, then it can go into a cell assay, and then it can go back, and then we go backwards and make new compounds, so the design, make, test cycle. So just 1 minute on this, and then I'll stop. If traditional high-throughput screening doesn't work, what will? Okay, that depends on your hypothesis about why high-throughput screening didn't work. And several of people's questions were about different libraries, how to choose your library, what different things are out there. So for example, in protein-protein interaction space, often high-throughput screening traditional methods don't work very well. And if our hypothesis is that the library is simply too small, 100,000 compounds or 1 million compounds is too small, or not the right chemical matter for my target, I want to use a much larger library. The main technologies now are virtual screening, where virtual screening libraries can be more than 1 billion compounds now. And DNA-encoded libraries. These libraries can also be hundreds of billions of compounds. If you have a way of measuring binding directly, you can use a DNA-encoded library. If your hypothesis is that the surface is too large for a small molecule, it's not a druggable surface, it's like a big shear wall instead of having big holes that you can put your feet into, then maybe you want larger molecules, natural products, more complex molecules, or macrocycles, or even biologics. And then finally, the third hypothesis is that the surfaces are complex with several small subsites. So it's not that it's a sheer rock face, but that the holes are far away, that to get my hands in and my feet in, I have a large surface to cover. There, the technology of choice is usually fragment-based drug discovery. And this is a technology where instead of making the whole molecule and then screening it, which is essentially high-throughput screening, you discover the molecule in little pieces. And then in a second step, link those into a high-affinity molecule. These are usually binding-based methods. And I was going to give you an example of just, okay, last point. Drug discovery, as I said, is really a team sport, and so different funding agencies, CPRIT and NIH, have different ways of trying to help you do that kind of team science. And the way the National Cancer Institute has done it is through this chemical biology consortium, where there are several groups around the country that are essentially contracted to work together on projects. And one project that we have came from the Shea Lab. He's now at Washington University. His idea, he discovered this target. He thinks it's going to be really good for cancer. He's an oncologist. He doesn't want to do drug discovery. So he brings it to the NCI through the NEXT portal and asks us to develop a project team and works with us to develop those molecules. And so these 4 teams in red worked with James's lab, and he brought in a collaborator, a crystallographer from Columbia, and together we've been prosecuting this really difficult target and have first-in-class molecules for this target by working together as a team, and the collective experience of it's a fragment-based drug discovery project, working together on that. Okay, so a drug is the intersection of potency and efficacy, which we call pharmacodynamics, ADME and toxicology, which we call pharmacokinetics, safety and specificity, which you think of as safety, selectivity and specificity. So you want to thread that needle. And this is just a positive spin on what I said before. So select the right targets, validate them properly, select the right assays, and focus on the therapeutic index, and you'll be just fine. Okay, I just want to end with some resources for you. And thank you very much for your attention.